Welcome to this explainer. Today, we're taking a microscopic tour into the uniquely complex and honestly fascinating world of medical mycology. We're going to uncover the secrets of the shape-shifting organisms living in, on, and literally all around us. If you've ever wondered how a simple organism can thrive in the dirt of your backyard and then perfectly adapt to survive inside the human body, well, you're in for a treat.
Here's our roadmap for this explainer. We'll start out with the fungal kingdom, compare fungi to bacteria, look at the anatomy of a fungus, explore the fungal fortress, see how we actually go about identifying fungi, and then end with their incredible shape-shifting forms.
Section 1. The Fungal Kingdom. First things first, we've got to completely shatter the misconception that fungi are just some type of weird plant.
Fungi actually have their very own, entirely distinct kingdom of life, Mycete. Why? Well, unlike plants, they're absolutely not photosynthetic. They have zero chlorophyll. And they don't use cellulose to build their structures either. Instead, these eukaryotic organisms survive as saprophytes feeding on dead matter, as commensals just peacefully hanging out with their hosts, or as parasites. They are incredibly successful survivors, and they play entirely by their own biological rules.
Section 2. Fungi versus bacteria. To really wrap our heads around what a fungus is, let's compare it directly to something we're all a bit more familiar with: bacteria.
The complexity gap between these two is absolutely huge. Fungi are eukaryotic organisms, meaning they're actually much closer to our own human cells than simpler prokaryotic bacteria. Just look at the size difference. A typical fungus is roughly 4 micrometers across, while a bacterium is about 1. But the real kicker is the architecture. Bacteria have a peptidoglycan cell wall and no sterols in their membrane. Fungi, on the other hand, build their walls out of a super tough substance called chitin, and their membranes are packed with sterols. This fundamental difference is really the bedrock of medical mycology.
Moving into Section 3. Anatomy of a fungus. Let's literally zoom in and look at the biological machinery operating inside a single fungal cell.
Looking inside, it's kind of wild how familiar this looks, right? You've got a defined nucleus holding multiple chromosomes, energy-producing mitochondria, an endoplasmic reticulum, a Golgi apparatus. These guys are fully equipped metabolic factories, complete with the exact same membrane-bound organelles found in our own mammalian cells. They are incredibly self-sufficient.
But here is the why it matters moment for medicine, and it lies right at the boundary of the cell. Our human cell membranes use cholesterol to regulate permeability. Fungi use a slightly different molecule called ergosterol. Now, that might sound like a tiny chemical detail, but in the medical world, it's a total game changer. It gives scientists a highly specific target for antifungal medicines, meaning we can attack the fungus without accidentally wrecking the human cells around it.
Section 4. The Fungal Fortress. Just outside that delicate ergosterol-lined membrane is one of the fungus's most resilient features, its cell wall.
You really should think of this cell wall as a highly engineered, rigid fortress. It's built in 3 main biochemical layers. On the outside, you've got mannan, which dictates how it interacts with hosts. Beneath that is glucan, creating these highly branched crosslinks that give the wall immense strength. And right at the foundation, chitin. It's an inert, completely insoluble polymer that acts as the ultimate structural scaffold. When you see it laid out visually, that thick layer of chitin makes the fungus unbelievably resilient. It can literally shut out toxic threats from the environment. This highly structured, layered shield is a totally different ballgame compared to the simpler peptidoglycan wall of a bacterium you see below it.
Section 5. Identifying fungi. Because these unique fortresses are microscopic, lab scientists have to play biological detective using specialized chemical stains.
Scientists smartly exploit that unique wall biochemistry we just talked about. Suspect a fungus? Hit it with calcifluor white, which specifically binds to the glucans and chitin, making the fungus actually glow. Or use PAS to dye the polysaccharides a bright magenta. You can even use India ink as a negative stain to outline the sneaky, hidden protective capsules around specific yeasts like Cryptococcus.
And here is what that detective work looks like in action. This GMS stain has targeted the fungal wall, turning those invasive, branching structures a stark, unmissable black. It makes the pathogen pop right out against the lighter background of the human tissue. It's a brilliant example of using the organism's own armor against it to get a clear diagnosis. Finally, Section 6, Shape-Shifting Forms. Let's zoom back out from the biochemistry to see how these cells physically arrange themselves in the real world.
Fungi generally rock 2 main physical forms. First up is yeast. These are solitary, single-celled organisms, usually just 3 to 15 micrometers across, taking on a spherical shape. And their reproduction is super straightforward. They just pinch off a new copy of themselves in a process called budding.
You can actually see that exact budding process happening right here. Look at the larger parent cell right in the center, and perched on top, that tiny new bud breaking away. It's an incredibly efficient way for them to rapidly multiply when conditions are right.
The second major form is mold. Mold completely ditches the single-cell lifestyle. Instead, it grows outwards as long, multicellular, branching tubes called hyphae. Just think of them as continuous tubular extensions enclosed in thick walls. When these hyphae actively grow, they weave together into an incredibly dense network called a mycelium. You can see the microscopic branching on the left and the overall structure on the right. Fun fact, when you spot that fuzzy mold growing on a piece of fruit, you're actually looking at one of these massive interconnected mycelial networks with the naked eye.
Which brings us to, arguably, the absolute coolest survival trick in medical mycology, dimorphism. Dimorphism literally means two forms. Certain fungi can actually switch between being a mold and being a yeast. But what exactly triggers that shapeshift?
It's all about their environment. Things like pH levels or carbon dioxide play a role, but the huge one is temperature. Out in the cooler soil at around 25 degrees Celsius, they live as wide-reaching, branching molds to absorb nutrients. But the second those spores enter the warm, 37-degree environment of a human host, boom, they shapeshift. They instantly transform into a single-celled yeast, which is way better for traveling through the bloodstream and dodging our immune system. It's a terrifyingly brilliant pathogenic strategy.
And that wraps up our micro tour. We've seen how these fungi build indestructible chitin fortresses, how wildly different they are from bacteria, and how they dynamically shapeshift just to survive. Knowing how perfectly adapted they are really leaves you wondering, what other crazy survival secrets are hiding in the vast, uncharted fungal kingdom all around us? Thanks for hanging out for this explainer, and keep questioning the micro world around you.